morning. We might have more people joining, but we're going to start. And the name of this panel is Citizens' Science Approach to Searching and Curating Literature on the Effects of Spaceflight on Cardiovascular Outcomes in Rodents and Humans. Our speaker today is Matthias Nasset, a graduate student from McGill University. And I'm going to give the floor to Matthias.
Thank you for that introduction. I will just quickly start sharing my screen and get my presentation all loaded up. All right, so let me just get a laser pointer here. All right, so thank you everyone for tuning in to my presentation today about the citizen science approach for searching and curating literature on the effects of spaceflight on cardiovascular outcomes in rodents and humans. So to begin, I want to just go over an agenda of what I'll be talking about today. So I'll give a little bit of background about myself and my training and why this is relevant. Some background about space physiology and literature reviews. Particularly, this will be focusing on some previous bone outcomes that we have worked on. How this data can be reused, and accessible, and then additionally how there are other system linkages that can be linked to this. Next, I'll talk about the systematic review and meta-analyses of cardiovascular outcomes in space. This is the project we're currently working on, and as I'm sure you know, or you recognize by the title, this is kind of the focus of the presentation. Then I'll go into a little bit of the citizen science approach that we used. For some of you, this might be novel, a new idea. Others, this might be relatively familiar, but I'll talk about this and how we actually implemented this into space physiology research. Next, I'll talk just about the screening results we had. So this is using the citizen science approach. I'll talk about where we were successful, where we were not, and everything else in between. And then finally, I'll talk a little bit about some of the outreach programs we've had develop out of this that have gone above and beyond what we originally planned, but are definitely contributing to enhancing this program as we continue along. So, like I said, I'll give you a little bit of background about myself. So I'm actually an engineer by training. I did my undergraduate degree in mechanical engineering, the biomedical option, at the University of Alberta in Canada. During this time, I was a part of many groups, some of which include the Undergraduate Astronomy Society, AlbertaSat, a group where we actually ended up building a satellite and launched it to space. And then additionally, another project I worked on was an artificial larynx capstone. So as you can see, I've got quite a bit of background just generally working in space and health-related work. Additionally, I also have 4 years of research in a neonatal baby lab. This is where I worked on medical device development, including resuscitation masks and real-time force and pressure sensors, and additionally, medical technique improvement in the realm of chest compressions and oxygen delivery. So this isn't necessarily my first introduction to research, but rather an extension of the work I've done before. So this brings me to current. Where are we now? So like I was said in the introduction, I'm a third-year PhD student at McGill University. I'm actually in the Faculty of Medicine and Oral Health, or Dental Medicine and Oral Health Sciences. This is a bit of a long story, but long story short is there's still engineering work being applied, so I'm kind of fitting right in. And today we'll be talking about my project, which is focused on cardiovascular health during microgravity. So moving on now to the bone outcomes meta-analysis that I had previously mentioned. So my lab had actually done a few years ago a meta-analysis on bone outcomes in humans and rodents in space. So these as experiments in space are costly and rare, legacy data that can be acquired over years of space research are important for enriching data repositories and completing high-level conclusions. So what this means is that it's very important to have the data accessible, and not only that, but reusable, so that you can be able to draw larger scale conclusions from these results. This is very beneficial, as many of the spaceflight experiments, as like I said, they are costly and rare. So most of the population sizes tend to be quite low, and you can have difficulty obtaining quite a large variety of outcomes. So having them being able to pool them together and draw these larger conclusions for advancing our knowledge on spaceflight is very important. Particularly, I want to reference three papers that our lab did produce that focus on these results. The population groups are a little different. So here we have Stavnishuk et al. 2020, which focuses on the human side. And then we have two papers here, Fu et al. And Goldsmith, which focus kind of on primates and rodents and more animal model-based results. So starting with the bone results in the humans, The main things that came out of this are that the changes in the bone were skeletal site dependent. So what this means is different areas of the bodies actually experience different changes. Particularly, there was more bone loss in the lower body and the pelvis and legs compared to the upper body. Now, if you look at the image on the left here, you can see this being quite clearly illustrated by these results. So as you can see, the upper body here maintained a relatively stagnant change across a very long mission duration of about 300 days. There's very minimal change. However, when you look specifically at those areas that we had mentioned, the lower body, pelvis, and legs, you can see that there was a dramatic shift in bone density across this time. Additionally, this can be extended to the actual specific bones in this lower body, as even one example of just the heel bone experienced significant decrease in bone density. This is further emphasized by these graphs on the right side here. As you can see, once again, once you focus on the upper body, there is a very minimal shift on the average of all the studies from the no change line. Additionally, when you look at those areas that we had previously mentioned, like the pelvis, lumbar spine, and the lower limbs, you can see that the shift is much more significantly to the negative side, meaning that there was a decrease in overall bone growth. Now, moving on to the animal studies. So while these results were shown in humans, there are much more other studies that can be analyzed looking at animal models. So here, as you can see, we have very similar results. As in trabecular thickness, you can see there's significant bone loss, decrease over time, and same both in rodents or rats and mice. Now, the area that I wanted to focus on more is this image on the bottom. As you can see, once again, they kind of split the rodent skeleton into several main groups. Again, kind of focusing on that idea of upper body, mid body, and then like lower body as well. And once again, you can see here that there was significant difference between those groups, as every time the lower body was pretty much drastically more shifted into the range of whichever caused the larger shift. Now, one thing I want to talk about now is the actual time required to complete these meta-analyses. So the first one, the human study, required 3 years to go through about 6,000 articles to the database, put them all together and do statistical analysis and then draw all the conclusions. The second paper, which was the Fu et al. One, which was the rodents, required about 2 years of study or 2 years of completion time for approximately 14,000 articles. These can be proportionate to either it's like team size, there's expertise on the team, there's many different variables, so which is why you can see different numbers might not directly correlate to an exact time. However, the big thing that I want to focus out of this is that they do take quite a bit of time. So 2-3 years for studies of this size are needed. Additionally, although these studies are quite lengthy, they are a great mechanism for involving undergraduate students with space biology projects and allowing them contribute meaningfully. They also allow the students to kind of start exploring not only research at a graduate level or further research levels, but get the experience of working in a lab in that way. So one thing I want to talk about now, additionally on top of that, is the data reuse side of the meta-analysis itself. So generally, most of the time, you'll have the aggregate data published. So what this means is you'll have grouping of animals or humans or whatever sample you're using for relevant statistical analysis. This just makes it easier to easier to read when you're publishing and easier to visualize, especially in some of the figures. However, there was quite a significant amount of work that was done putting all of this data into tables and organized primary data, as you can see. This is a very small snapshot of what is thousands of different lines in Excel and columns of just data going in of the various groups that were analyzed in our meta-analysis. So then we were kind of stuck. We had all this data nicely organized, all set and everything. We were wondering, well, what can we do with it now? Like it's just kind of sitting in the computer. There's not really much going on with it. So then we had the idea, maybe it can be reused or something. Maybe someone else can use this in a different way so that our work just continues to move forward. And we really don't want it to be, it's not necessarily for nothing, but all the extra work of organizing and making it look nice is just used again. So this is where this new kind of experiment or archive database is coming in from NASA. So as you can see, NASA, or I'll start a little bit more about NASA. So previously they had a database called GeneLab where they would where you could upload omics data, but nothing necessarily associated with physiological outcomes. However, within the past year or two, NASA has begun to develop a new open science data archiving system called the Open Science Data Repository, or as you can see on top, OSDR. So this is a open access database where anyone can access the data of just all spaceflights of any spaceflight experiment that has really been conducted. I say any spaceflight experiment, but this is, like I said, a growing database. So currently there are both experiments that are being uploaded, and there's a lot of work on previous experiments being uploaded, curated, and archived properly. So I have actually been uploading some of the data from those bone meta-analyses that we have. So if I quickly just go back, so all of this data here, I've effectively been truncating it and putting it into much smaller files just to have it more accessible, and then uploading it into this open science data repository where anyone can download this data and reuse it. Additionally, they have overarching description processes, which describe the abstract of the article that was published, the experimental methods, many different things, which I'll actually get to in the next slide. So looking at some of those other things that I was talking about, they had two other categories, namely called the assays and the configs or configurations. So what these are is these describe the work that or how the work was being done. So this generally falls under the methods kind of area where it allows people to see how exactly those results were obtained. And if say you wanted to reproduce it or reproduce the environment or methods, but just hoping to get different results, this allows you the freedom to do that. So as you can see here, we focused on two main assays for the bone results from our study, which were micro-CT and bone histomorphometry. The example you can see here is micro-CT or microcomputer tomography. So this data set here effectively, like I said, describes the methods that were used in the micro-CT imaging. So you have various information, such as the exact model that was used for the scanning, what medium was used, the specific location, the sizes of the scans, and so on. This actual set of data is quite large and it would take me several slides to go over. So I just thought I'd give you a quick little snapshot of what this is. But effectively, I just want to show that it is very important to not only have the data that you have itself written up, but you need also descriptive methods for describing how the project was completed and other information such as descriptions on the samples, as there are other separate sections for specifically just the sample information, such as what kind of models were used, the age, flight duration, and so on. So moving on now, I want to talk a little bit about bone and cardiovascular linkages, because you might ask, oh, why is my project title on cardiovascular outcomes? However, I've only been mainly talking about bone for now. So I want to say that 5 to about 11% of the cardiac output is actually supplied to the skeleton. As you can see here, the bone has many different arteries and veins that feed into it, both providing growth and providing nutrients. So this bone environment can actually be locally altered by cardiovascular changes in microgravity. These changes can include fluid redistribution, which is some of the most common, some alterations in blood pressure, and even changes in cardiac output, as you can see. So a decrease in the cardiac output could result in decreased blood to the bone, which may have some kind of impact on the bone growth itself. Intuitively, these linkages are also kind of there, as blood is fabricated in the bone marrow, and so you can already kind of tell that there's this kind of heavy interrelation between the two. So this brings me to the motivation behind the study that I'm describing today. There is a relative lack of large-scale reviews for cardiovascular data. So primarily, there's just a lot of individual studies done. And like I said, one of the big issues with that is that the sample sizes are generally pretty small. So this can be hard to generalize to larger populations, and especially with the amount of people who are pushing to go to space these days, it's very important that we have a thorough understanding of the processes and of the interactions within the physiology systems of our body. So we also wanted to look at, is it possible to connect this data between missions or samples? So like I said, we know that there have been reports of bone and cardiovascular linkages. So we wanted to see, is there some way that we can find bone and cardiovascular changes in a particular subject? So what we mean here is more often than not, if there is an experiment going to space, say there's an astronaut group going to space, you'll have several different groups of experts working on different projects of experimentation on them. So what this mean, or so in one way, you could have one group looking at the bone results or bone decay on the astronauts, and you could have a complete other group looking at the cardiovascular outcomes, while it's all done on the same sample per se. So we're hoping to look and trying to find, at least either on a mission level or a sample level, some commonalities between these two, so that maybe we can start to directly connect the results. So maybe what we might see is if the bone volume is decreasing, there is, like we said, maybe decreasing cardiac output, and we can start to correlate these values together across multiple studies. But what exactly we're looking for at the moment, we don't know. And because of this lack of cardiovascular data, we're starting kind of from the back end of it, building up from our knowledge base on the cardiovascular side, and then hoping to connect these later on. So to start this, what we did is we went to the AWG group. So what we did, I'll talk a little, go into description about what this is in a little bit, but what we did is we presented this cardiovascular, that's what CV is short for here. We presented this meta-analysis idea to a group of experts. This experts came from the NASA Ames Life Science Data Archive, or ALSDA, Analysis Working Group. So this is a very large group that meets monthly just to discuss various processes around space research and space experiments. So this can be in the realm of, enhancing experiments, just relative ideas and topics going on, or one of the main focuses right now is enhancing the OSDR portal and making sure all of the methodologies, assays, and configurations are accurate so that they can be reported correctly. So from our presentation, we had about 25 experts from around the world join to consult on this project. This number is still growing as we have a number of experts who continue to join as our project grows. But they were primarily involved with the project conceptualization. This is in the realm of just kind of the ideas on how to approach this project and the key term development. As you can see, some of these key terms that were devised here for our search. And additionally, they continue to work along as project reviews. So they will sit in every so often. We'll check in with them just to see how the project is going. If we have any particular questions about the subject, we can go to this group of experts to have their input as well. So now I'll talk about the search results we had. So we put all of these terms here. I mean, this is a very truncated and short version of what we had. It was much longer than this, but to show that it would be impossible. So we put these into a search. We had librarians execute the search for us, and we ended up receiving just under 20,000 articles. So then this process, as you can imagine, this is larger than both of the other studies I had previously mentioned with the bone. So something of the size might take several years, probably in the range of 3 or 4 years to complete. That being said, after the duplication, we had about just under 19,000 studies that were screened. As I'll get to with our methods, we ended up completing this and getting down to about 3,500, which are currently being worked on for full text screening. The main thing I want you to get out of here is the number of articles that we decreased from. So from just under 20,000 to 3,500, and keep this 3,500 number in mind for later. So you might be asking, how did we go through this large volume of studies relatively simply? So what we did is we actually had an idea because we had the experience with the projects before, seeing how long they took, seeing how undergraduate help is both beneficial for them and us, to look at recruiting public help to advance legacy data findings and curation. So this is one group that particularly found this useful. So we term this as the citizen science approach. So this methodology is actually commonly used in astronomy and geology. This is where there are large data sets, anyone can contribute. So particularly, for example, in astronomy, what people will do is they'll take their telescope out, they'll look at a certain area in the sky, maybe they'll see a new star, they'll document this somewhere, and then other people can verify it, and that's how you kind of increase your database. So our approach, I just want to mention here, is actually recognized as the Solstice approach. It is called the Space Open Life Science Team for International Collaborative Exploration. So we did our recruitment. So one of our collaborators, he spearheaded the recruitment. We focused on organizations with underrepresented groups in STEM, particularly the group SEDS and the Space Generation Advisory Council. And to our surprise, we actually had over 100 people join in just 1 week of recruitment. And we had to close it down actually at that point because we didn't want it to get too out of hand. So our participant educational background. So as you can see, the demographics for the people. So we had of these 100 people, they hail from over 28 countries with, you can see, various densities from around the world. And their backgrounds were quite spread out, with you here being the undergrad. So about half of the people were undergrad, a quarter were graduate students doing their masters. We had some students doing their doctorates. We had some postdocs, and then we had some people from industry all join. So as you can see, we have quite the background of young professionals working on this project. Looking at how we approached the education side of this project. So what we did is we gave weekly 1.5-hour lectures on these various topics here, which include project background and methodology, space physiology, experiments in space, and literature reviews. Additionally, we provided screening workshops, and we also maintained weekly office hours where students could come in, I said students, but we actually moved away from this term and go young professionals now, because as you can see, not all of them are students. And we provided weekly office hours for them to join and ask questions or get clarification. So looking at the screening productivity of this project, so initially after 2 months, we had about 10,000 articles completed. So you might wonder why this is at 40,000. This is just because for the purpose of the project, we had two people screen each article just to reduce overall bias and reduce the error. So after 2 months, we had about 10,000 articles screened. We noticed while it is great progress, we thought we could be doing this a little bit better. So what we did is, actually, after having screened some of the library myself, my supervisor and I went back to the students that we gave the initial lectures on and gave them updated information about how to screen. This came in the realm of particularly some of the tough terms, mainly focusing on flight, the word flight, because flight would come in and the people would get confused whether it was actual space flight or just a commercial flight. So we clarified this, and then within 2 more weeks, we actually ended up completing 30,000 more screening articles. So this kind of a volume of studies would usually take around 1 to 2 years, but we were able to get it done in at most 2.5 months. As you can see, this is just the screening distribution. So the McGill teams, this was me, my supervisor, and a graduate student we had working on the project. We did a significant amount of the work. This, again, falls into the realm of us being able to make these changes and then provide updated lectures, while the rest of the work was quite spread out amongst the rest of the people. Here's a quick description about the screening distribution. So as you can see, it was split up into various groups. Generally, most of the studies were in English. They were on humans and rodents, mainly focused on simulated and actual methods. A variety of methods for simulated were used, with the general focus being on cardiovascular and primary articles. Moving to the full text screening. So this is the final results of what we ended up having. So we have various groups that we wanted to look at. We have humans in actual spaceflight, rodents in actual spaceflight, humans in simulated spaceflight, rodents in simulated spaceflight. Radiation we included at the start, but we decided to exclude this for later purposes, as there's a very large area in itself that we needed to focus on for this. And then other were ones that just didn't fit into these categories. For the purpose of what I'm going to be talking about today, we are focusing on human and actual spaceflight. So this is what all of our efforts are being put into right now with the project with the hopes of completing this meta-analysis. So what do we plan on doing when we're done? So once we're done with the full text screening, we plan to look towards data extraction. And what we plan on doing here is we have, while the screening is being done, we're labeling each section with these titles here, and these allow us to organize and group the outcomes of each paper. And as you can imagine, this is going to be very useful when it comes down to looking at implementing these cardiovascular outcomes into those assays and configurations in the OSDR portal that I had previously mentioned. Like I was just saying, we do need to implement these into the portal, and it will allow us to maintain unity across the extracted results and allow it to be reused much more easily. One final thing I want to talk about that's come out of this project is we actually ended up having a high school outreach program as well. So not only just reaching university level students, but we had some high school level students join the project through NASA's AIML High School Students Program. These students participated in weekly lectures about space physiology experiments. They did workshops on how to read research articles, where we had discussions about results and findings. The goal of this for us was to have them work on the reviews as a check. So what they would do is they would read the reviews that we had included, check the references of those reviews to make sure that the articles being referenced were included in our original studies. As you can see here, they did a variety of work over an 8 weeks program. They focused on 3 particular review articles where they looked at, like I said, the references from those groups. We additionally had very positive feedback from the students, where even I learned some new ways to describe the program that we're doing. So some of the limitations of our project, it is difficult to implement rolling acceptance for a citizen science project. So what this means is it's hard because of the nature of the project. We have people asking to join all the time, but it is hard to accept someone who is brand new and just throw them into the full text screening process. Not one, because they're less familiar with the project itself, and 2, we want to reduce the overall error that it can be made because project familiarity is important. However, we do find other areas for them to contribute in, such as, like I said, with that high school program, they can work in the reviews to help us make sure that everything is being done properly. Additionally, we want to recommend that labeling the articles should be done during the screening. In our initial phase, we labeled the articles after the screening. This provided a little bit of downtime for the citizen scientists, which can kind of sway them away from the project. You know, they might be either forget about it, or because they're not working on it in a little while, they can move away from this contributing as much as they did and using them to their full potential. And finally, the biggest bottleneck of our project right now is that the full text articles are difficult to obtain. As our studies are looking at every spaceflight article ever published, so this is going back all the way to the 1950s, it is quite difficult to get these. So like I said, this is increasing the downtime for the citizen scientists, which we're trying to decrease as best we can. However, we found a little bit of a workaround by using multi-institutional access. So by using these citizen scientists and their various locations around the world and various institutions that are associated with, we've added the full text article obtainment process significantly to help push this project forward. So looking at some of the implications this might have, so very clearly there's the research acceleration, as this decreases the amount of time required to do a review of this size.
Matthias,
yes.
we have 1 minute left, so let's wrap it up, and then if there is a question.
Okay, perfect. Yeah, this is my last slide. So yeah, it improves data curation, as you can see in the portal, promotes open access databases, provides unique experiences in space research, promotes scientific research as a whole, and has a multidisciplinary approach. So I just wanted to acknowledge some of the people I worked with in my lab and across the institutions. Just here's my references, and thank you.
Thank you. If anybody has any questions, please type them in the chat or in Q&A. Okay, we have a question.
So Charles asked, why is it difficult to obtain literature from the past? So mainly just because of the actual archiving processes that have been implemented before. So for example, the older the articles, it's actually harder to, like, they haven't been archived properly. So we don't have the direct data from those. Additionally, it's more difficult to reach the PIs, as often the principal investigators on those projects are either retired or moved away from this field. And one final other big problem that we've run into with the literature from the past is just differences in reporting. So even like just methods are significantly different, which have to be worked and standardized, units, and so on. Yes, that's true. It depends on the journal.
Thank you. And thank you for your question. Thank you, Matthias, for a wonderful presentation. It's time, so we are going to end the session, and we have panels going on throughout the whole day, so please check them out on our website and join more. And thank you, Matthias. Bye-bye.
All right. Thank you. Bye.